Hi Hires, we are looking at the DNA in the Genome Unit Key Area 4 cell differentiation. Okay, so this is the final result of all the transcription and translation stuff from the last key area. Every cell inside you does not look the same, but you know that they all have exactly the same code. So if they have exactly the same code, then there must be a difference in how that code is expressed. So the pattern of expression, the timing of expression, all of that will make a difference in what actual cells you get. Okay, and that particular process, the process of moving from a cell which looks just a kind of neutral starting point for everything, all your cells at some point look the same, changing, that changes cellular differentiation. You're becoming different. This is just a picture of lots of different cell types from humans. So every single one of these cell types had the same code inside them, still has, apart from these guys down here because they don't have a nucleus, but everything else, the code is still there. So why do they look so different? It's because of the pattern of expression they have. So we have to know something specific about how we get from a cell which doesn't have differentiation to one that does. So the cells that can actually differentiate have particular names. Once it's differentiated, we can't go back. So once you've made a nerve cell, then that nerve cell is a nerve cell forever. It's not going to change to a heart cell. There are some, just to put in brackets here, we have actually managed to shift cells back from being not fully differentiated to ones that can now go to everything else, but it's not normal. It's not a normal thing. You have to do quite a lot of complicated things to the cells to make it happen. And what we have inside all living things is a very particular set of cells that can either end up in a loop of self-renewal, so they just keep on continuing to replicate, or they can differentiate and become a different cell. And they have particular names. In plants, these are called meristems, and in animals, we've got stem cells. Okay. So in plants, now you don't need to know as much about this as used to be in the course. So you will find, quite likely, in older textbooks, there will be a lot more information on this. They will talk about something called cambium, and you will need to be able to do it in quite a lot of detail, but not now. So if you see anything about that, just don't panic. The only thing you need to know is that in plants, there are meristems, are groups of cells inside the plant that can either divide, that's self-renewal, or they can differentiate into other cell types. Okay, where you find them, it's kind of obvious. You find them at the point where a plant grows. So in this case, we've got, this would be at the tip of the plant, so we'd expect root and shoot would be where you would get them. And we also have them at the edges. So that would be if you have a plant which is getting wider every year. So the rings that form in plants every year are due to meristems. Okay, so just the term meristem. You need to know more detail about stem cells. Okay, so your stem cells are in animals. And what we're looking at here is, again, cells that can either self-renew and can differentiate. So there are two options. But we also look at stem cells and we see how many different things can they actually differentiate into. And that splits them into embryonic stem cells and tissue stem cells. Your embryonic stem cells can actually be split two ways as well. And the first one, actually, I'm going to tell you, but you don't actually need to know much about this. Okay, early doors, when you've just had a few divisions have run in the embryo from the zygote moving forward, you get cells that could become anything. Okay, so they're called totipotent. They have total potential, total capacity for changing into anything else. And by anything, we mean every single possible cell type that is in every part of your body, plus it could also become the placenta. And placentas, we don't have that past this stage, because after this you have no need for it. Once that's made, you use it for 9 months and then never have to grow placental cells again. So actually it's not this really early one that they need. This is the bit they need. Okay, they need to go a little bit further on, okay, to pluripotent cells. So at this point we can still switch on and off all the genes and we can differentiate into any cell type but not the placenta. And these pluri meaning many potential or many capacity. And so this is early embryo and these are embryonic stem cells. They are the ones that are most useful from a modelling and potentially therapeutic point of view. Next one. So if we keep going on and we get to the point where we're no longer in an embryo, but you now have an adult with lots of different localized tissue, we can still find stem cells. But what you have now are multipotent cells. So that means they've got many potentials. They can't differentiate into everything. They will develop into cells that are like the ones that they came from. So if you have something that's come from bone marrow, which is a very common one as an example, bone marrow can differentiate into any blood cell type, but it can't differentiate into like heart or brain. So these examples at this side would be saying if you have brain stem cells, they will be able to differentiate into different forms of brain cells, but not into heart or bone. You know, so it's a very specific set. And you get them in lots of different places. So the first use of stem cells would be looking at a therapeutic use. And a therapeutic use is where we're using them medically. Okay, and because they can self-renew and they can grow for longer than normal cells do, as long as you give them correct conditions, you can actually grow these quite well in a lab. So they're already being used in several areas, and there's lots of clinical trials that are being done with stem cells. The two that you're expected to kind of see in are in the content set: skin regeneration and corneal repair, so the front of your eye. But basically the potential is for any place where you have damage to cells that have not been replaced when they otherwise would have been. So maybe if you're talking about something osteoarthritis, excuse me, then that's looking at cartilage at the end of your cells. If you're looking at any form of damage potentially to brain through any of the degeneration diseases that affect that, maybe that could be a way to go. There's so many different potential therapeutic uses. So here we have an example of trying to use an embryonic stem cell for a therapeutic use. In particular, what you're trying to do is to add back in retinal cells which have stopped working as well as they should. And you'll notice that we have this up here, this IPS. This is an induced pluripotent stem cell. So this is one of the ones that has all of the functionality of an embryonic stem cell, but actually was not from an embryonic stem cell. This is the ones where they've managed to kind of push them further back in their pathway. So we have the culture, you grow the culture, you then give it all the necessary cues. Now, so this, knowing what these are, that's not easy. But once you know that, once you know what the signals are you need to do, then you get differential expression, and once you've got the expression, you get differentiation into different cells, and then you can inject the cells. It shouldn't surprise you that the eye is one of the places that a lot of the starting things have been at, because the eye is easy to get into. So things like skin, eyes, lungs even, these are all good surfaces to try because you can get into them. If you're saying, right, okay, we want to look at kidney, liver, that's harder because you have to get into the body to do that, whereas you can kind of apply these quite easily. So we then have the other major thing that stem cells are potentially useful for, and they are very useful as a model system because they are cells that you can grow quite well and keep going for quite some time. So you can test drug systems, you can see how they respond to different diseases, and they're really useful to try and track through cell processes, so trying to figure out exactly how a particular chemical is changed inside the cell. So you're looking for the metabolic pathway. And it is a larger thing, and it's becoming more of a thing. Ultimately, though, there is a huge ethical issue with this, and that can't be dodged. Okay, the point with embryonic stem cells is that those embryonic stem cells involves the destruction of embryos, which is why that induced pluripotent stem cells may become even more of a thing. But it is a question that has to be weighed up, and it is something that you cannot avoid looking at. For a lot of things to do with biology, there are ethical considerations that should be looked at and should be considered. Okay, and that's cellular differentiation.